Morning, everybody.
Good morning, Angela.
Good morning. Good morning, Angela. Nice to meet you.
Good morning, Teresa.
Teresa. Hi. Well, finally, I managed.
Yeah, I also had some technical difficulties right now in the.
Yeah, I registered myself on the Global Summit, but it took me some time, but now it's okay.
Yeah, very good. So. Maybe we wait a few more minutes until it's really 9 o'clock.
Yes.
Angela, you gave a very good overview. It was really inspiring. So good to hear.
Thank you very much. You will provide the concrete, applications now. Yeah.
But also the progress already reported, that was really nice.
I couldn't get to get into that plenary session today. I tried. I guess probably my registration was such that it was only for this workshop, but I couldn't get through the plenary session today, other session today, this morning.
Now I was kicked out. I mean, I made a mistake. I pressed the wrong button. Then I was kicked out, and I couldn't enter again anymore. I think it was the plenary was full. I just landed in the in the waiting room. But yeah, I don't know. It's yeah.
So I missed your presentation, Angela.
Yeah, but it's
maybe you can give a very short summary.
You can have a look then at the recorded plots, but essentially I was introducing the audience to the microbiome support activities and just providing a very rough overview of microbiome applications in the food systems, but only at the from a very above level, let's put it like this. And I'm expecting the speakers now to provide more details, which we can further discuss.
Yeah.
So maybe because we will soon start, just a reminder to maybe turn off your video and also your microphone if you're not speaking, and only turn it on when you're speaking. So I welcome everybody. Thank you for joining us in these breakout sessions. So the breakout session, microbiomes for the bioeconomy, hype or hope. You have here some recommendations what to do. So the session will be recorded, so the speakers are asked to share the presentation through the share button. Please keep in time. Turn your microphone off unless you're speaking, and also your video. Here are some. We can have a chat, which we can then use. So you have a chat function. So please enter your chat and your discussion points here. Okay, so then our first speaker will be Lene Lange from Bioeconomy, LLA Bioeconomy. Her talk is about microbiome functional annotation provides basis for gut health, promoting food and feed ingredients and feed-induced methane reduction. So Lene, the floor is yours. You are muted. You're still muted.
Thank you so much. And I'm sharing my screen. And is it working all right?
Yes, it's you just have to turn on the presentation mode, but we can see your screen and we can hear you.
I am in full screen or not?
No.
So which one is your screen sharing? Which one? I forgot where it is.
No, you just go. It's usually on the bottom.
Yeah, but I already did it in my PowerPoint view. I already did it. And it doesn't function when I come into here. Can you, otherwise I take it from here. Is that okay?
Yeah, okay, it's okay.
Okay, because time is short.
Yes.
Great. Thank you so much, Angela, for introduction and for giving me this opportunity. I will say two things. First, it is great to start looking at all types of microbes, not just the bacteria, and I'll go into that. And also needed in order to understand fully more the role of the microbiome function, we need functional annotation. So that's two things highest up. The first I will share with you is that I started at the before turn of centuries to look at the microbiome experimentally, and I looked into insect guts, and I found the most wonderful secretome-rich enzymes. But that was experimentally. Then we decided to work to go the DNA way, even though it is sometimes more difficult and take longer time to get a functional gene expressed than the cDNA and from the messenger RNA transcriptome route. But here, what I am really sharing with you on this figure is something I hope will be much more used, that you make a binning of here, it's an anaerobic digestion microbiome, where we have all the groups to the right on the screen, here to the different rights here. And then you have a functional prediction, based functional prediction of the secretome genes, of enzymes. Then we can actually bin and show which roles and functions the different types of bacteria are serving here in the anaerobic microbiome. So this one is a very important step forward to understand this part. Then we moved on at one part, and that was that we went into looking and refined our peptide-based functional annotation by making a new one called the conserved unique peptide patterns. And to the right in the slide, you can see that the basic thing is that we are sharing the different types, using all of dbcan, cdHIT, and all this to find the right protein. We then make a clustering, the cup protein clustering. And that is the point where we are clustering in the computer on a program all the proteins which are sharing specific patterns. And those patterns are worked out for each and every of the the carbohydrate active enzymes. Then the next part of this cup is that we are ending up in being able to predict the function, because we now have found out all of the recognized peptide patterns, and then we can group them and predict what function then have. The next and last on that one here is that we go on and are able to use this not just for single proteins, but for microbiome secretome. The next part here is the cup to show, and that is a complicated one, but see here, this is DH30, just one of the enzyme groups showing the principle of cup. We have a subfamily 5 here, and you can see that by the cup is divided in a number of cup analysis, dividing in a number of different cup groups. Subdividing subfamily means that we can have a group with only one function, so that with one characterized can actually share, so that we can predict the function. So in here, I'm in short say how we validate the cup based on this already published, your 2 papers referred to the left. And then it's also here on validated on glycohydrolase 30. And the concept is that we are possible to group according and predict according to the shared pattern, typically one pattern, signature pattern per type of function among the carbohydrate active enzymes, the caseins. Then it's also important to do 2 more things. I presented in this slide. One thing is to move into looking also the protozoa. We are sure, and it looks in preliminary activity, that we are really able to make, and the protozoa are playing a very prominent role in several types of, both in rumen, but also in gut cells. And therefore we start moving into finding out which enzymes protozoa have. This is just published, the first one in that effort. But if you just imagine that this one was in this presentation mode. You could have a total overview of a microbiome with the substrate it attacks, the different enzymes, the function, prediction, specific bounds of the enzymatic reaction it is attacking, and then how many genes of that sort, and then which family it comes from. So we have the family representing that function, working on that substrate. So with this overall, you have an impression of such a profile of a microbiome at a specific type of feeding. For this, so that's how, in order to give such a presentation, it's so clear when you do it like this, using this cup, where you get that full overview, which enzymes are there, what type of function they have, what substrate they work on. So we can use that then to demonstrate gut health effect of different feed ingredients, but also food ingredients. Here, so we can find the ones stimulating the healthy gut flora. And we document that prebiotic effect by changes in the organismal composition, but also in digestive function by predicting the function in the cup of the different types of enzymes. Then we can do the same thing. And that's what we are doing, working on right now in the Danish dairy funded project. And that is linking reduction in rumen methane to feed-induced changes in function of the rumen microbiome. And that is amazing what we have been able to find there. And it looks like we can find suitable feed ingredients which can lower significantly. What we are looking at now is something like 30–40% reduction. And even for some compounds, it can go down to much more, 80% reduced. I don't know how much we can upscale and how much can be used and good also for digestion, but it looks very promising. So organismal composition and digestive function combined with experimental measuring the methane reduction makes it possible for us to use the microbiome to actually identify such new types of feeding, which can make that kind of transformation of the microbiome function. So we can reduce risk of antibiotic resistance by finding better, more healthy prebiotic ingredients for the pigs. We can reduce methane emission from cows, and all is through the microbiome studies. And with the reduction of methane and the risk of antibiotic resistance, we are also at the same time finding such kind of ingredients which are being possible to develop from organic side streams so that we use all our food and feed resources much better and also get better health and better for the climate. Thank you so much for this. I hope it's not too hype, but I really, really think it's a great hope, a fantastic area in front of us. Thank you.
Thank you, Lene, very much for this great talk and very impressive applications. I ask everybody to put their questions in the chat. We will then address them all in the discussion after the short talks. So thanks a lot, Lene. Maybe you stop screening, stop sharing your screen. And it's my pleasure to introduce you to the next speaker, Brajesh Singh from the Western Sydney University, and he will talk about agriculture microbiome tools, a long road to success. So, Bra, the floor is yours.
Thank you very much, Angela, and good morning, everybody. I will, as Angela said, I'm going to discuss about where we are in what I describe as a long road to success in agriculture microbiome. I will share some of those slides that I recently presented at a FO meeting, around 2 or 3 slides, but hopefully that makes sense in this context as well. All right, okay. So why we are here and what the prospects, what our hope are. Currently, the microbial industry is one of the largest global industries, more than $1 trillion. And that does not include a lot of antibiotics and other medicines that is produced from the microbes. Agricultural biologicals currently sits around $5.5 to $6 billion. Out of that, 4 billion are the microbial in nature. the most exciting part of this research is that by 2026, it is expected the market of the microbial products, agriculture microbial products, will be $12 billion. And it's increasing at the rate which is a 3.5 times faster than the chemical pesticides. And in not very far or in very near future, the microbial products are estimated to be the similar size of a chemical pesticide industry today, which is around $55 billion. And this industry is growing and growing fast because of multiple drivers and success story. One of those includes the policy, top down. There is a lot of requirement from the different government agencies. European Commission's Green Deal is one of the example. And then the consumer demands for less chemical foods. The European Commission's Green Deal requires 50% reduction in the chemical pesticides by 2030. That will be expected to be replaced by the biopesticides. And there, as I said, there's significant investments and excitements from both the industries and government agencies and everything else to drive this industry because of the multiple benefits this bring in. And there's enormous opportunity from the scientific point of view as well, because if you look at the human medicine, more than 50%, between 50 to 60% medicines comes from the natural products. Only around 11% pesticides comes from the natural products. So what it suggests, there is a huge lot of compounds there to be discovered, to be used in agriculture. So that's the promise. But it is also important we should know that the microbial products in agriculture is not new. We have been using that for decades. For example, we have been using Bacillus thuringiensis to Trichoderma viride for a number of years, or in fact, multiple decades. But it has not taken off the market as it was expected. And the few reasons includes the inconsistency in the field efficacy in these products. It works in one environment, in one crop, in one soil type, it doesn't work in others. There's a lack of product diversity. It is a very important we remind ourselves. If you look at the product, and this is microbial products in the market, it focuses mainly from the Bacillus genus, Streptomyces, and some here and there. And in fungi, Trichoderma, in fact, is the major one. So there is product diversity is quite limited to few taxa, and we need to go beyond. We heard in the first presentation, we also need to go beyond bacteria and fungi, look at the protozoa, look at the virus and so on. The other real challenges have been the sustaining the activity of the microbial product, from the fermentations to formulations to storage, and when it's applied in the field conditions. There are other factors that can also influence those sort of arguments. For there is a lot of product in the market, which is a snake oil, there is no product. And these sort of things has, in fact, constrained the commercializations and more products coming into the market. In the future, to tackle some of the issue, we need to move away from the single isolate approaches. That's my personal opinion. We need to go in the SynCom, the synthetic community approaches. When I say synthetic community, is different from the consortiums. If somebody wants to discuss that, we can have this question and answer to them, but it's not consortium. It's a synthetic natural combinations of the microbial communities. We need to go away from the just inoculant industry to look at our ability to activate the activities of beneficial microbe that is already present in the soil or already present in the plant microbiome. We need to have the tools to accelerate that activities. And then one of those tools is plant breeding, which is optimized for the beneficial microbes and genomic activities. So the key to achieve those hopes, those what I call is the future products, we need to have a sustained and long-term views to understand the fundamental knowledge of crop microbial eco-evolutionary interactions. It is simply not possible that we throw any microbes and expect them to work in all crops, all conditions. It will not happen. We need to target the crops, and then we need to target the microbes accordingly. We need to have a better understanding in applied sciences that when we put the microbial nucleus in the field conditions or any biochemicals to increase the microbial activities, that's what is the requirement of those microbial inoculants to colonize that particular plant species. At this moment, those are not defined. And we must keep in the minds there's a requirement for the multi-year research in the field trials. 1 year will not give you example. We need to run 5, 6, 10 years really to look what the benefits these microbial inoculants brings into agriculture productivity and environmental sustainability. There's other risk, there's important risk that we need to manage as we go. The product safety, also not just for the human safety, but also the plants and other plant species and animal species safety. The food safety is already important, particularly with the raw foods we eat like salad and the fruits. There's a social risk and the public perceptions and acceptance. This is a very good time for the microbiome research, but we make sure that we manage those public perceptions and not go into the same route what the genetically modified crop has gone to. And there is a regulatory risk. Registration and safety requirement need to be formalized. It needs to be aligned and need to be standardized across the globe so the products come to market. When it comes to market, gets to the individuals quickly. So in my last slides, what I'm proposing, if we're going to realize what the hope and the excitement the microbiome tools is proposing at this moment, we need to go beyond the single isolates and focus on the system-based approaches where we understand the plant microbiome interaction, eco-evolutionary interactions. You heard the terms before, the holobands and eco-holobands. And what are the requirements of the microbial inoculants to colonize the plants to express its phenotypes? We need to have a targeted crop microbiota. It's going to the first things. Some microbes are present in all plants of a particular species, and we need to target them because the chances of their colonization is higher. As a result of their efficacy should be higher compared to just throwing any microbial inoculants. We need a long-term public-private partnership from the receptions to the market for successful translational outcomes. And that also needs to coordinate the global approaches for most of those things we have just highlighted. We need a global approaches for the improved outcomes. What I'm going to end with this statement, we need a long-term visions rather than the short-term outcomes. So we need that government agency, the private company have a long-term plan with the resource allocated, not just for 5-year time period when the product is coming out, but what will happen after 10 years, 15 and 20 years. These tools have a huge applicability, huge potential, but we need a long-term plan to achieve that. Thank you very much for your time.
Thank you, Pra, for this excellent talk. Working in a similar field, I have to say I fully agree with all your statements. So thank you. Again, questions, please put them in the chat, and we move to our next speaker, who is Ulrich Schur from the Forschungszentrum Jülich, and his talk will be on microbiome technologies linking microbiome to crop performance. Uli, the floor is yours.
Can you see the slide?
Yes.
Okay,
Everything
thanks
is
very
perfect.
much. So I think it's finishing up the impulse talks, which you just heard, is a good kind of starting point for my talk. Essentially, what I'm relating to is technologies linking microbiome to crop performance, and we have heard a lot of great stuff from the microbiome level. If we want to put it into basis for crop performance, we need to go in the interaction with the crops. And microbes play a very important role in many different areas of the food system. And for the crop performance, specifically in the soil environment with natural soils as well as agricultural soils, with the rhizosphere as a very special range area of interaction between plants and the environment, plants in the microsphere. In the plant itself, the natural environments, but there are also crop habitats, agricultural systems. There's a lot of pathogen aspects where also microbiome as a whole plays a role. And also microbiome is important for the food and non-food sector. So from our plant perspective, the microbes play a role in the entire life cycle of the crop. So from the seeds to germination, to rhizospheres, to plant pathogens, to harvest, and to microbes on food specimen and non-food specimen as well. And we heard a lot of opportunities there in the last two Impulse talks. What we are interested is really to know how the interaction actually can happen. And this gives us a good opportunity then to design the interactions and like we just heard in the last Impulse talk, to have an interaction between breeding, management, and the microbiome as a whole. There are a lot of actions there which we can look for seed performance, for example. There's a lot of dressing points at the early stage of a crop, meaning the seeds. There are seed coatings where there are possibilities there for microbes being involved there, the protectors from biological systems. There are also seed stimulants which need to be applied there. Nutrient supply is obviously also an option there. And we have options to technology-wise, and you know that I gave a presentation just in the plenary before about that digital technologies, and these digital technologies offer new opportunities. For example, we have what we call PhenoSeeder, which is a system which gives us the opportunity to look at individual seeds and do analysis there. So we can do a lot of 2D and 3-dimensional traits, which give us information how these seeds are performing. There's a lot of seed microbiome interactions. We can look into seed quality, at the sorting. We can look at coding issues, how they're actually working, and how distributions of microbes are actually on these biologicals are how they are on the seeds. This is how a system looks like. It's an automated robot which delivers this kind of information. It's not sure if you see the video running, but it gives us a lot of opportunities which we can do with seeds and how they can actually work with all these different traits. Beyond that, obviously, the germination is very important, and we need to work for the germination in the environment, which is really where the microbiome is actually happening. For example, in the real soil environment, there are also increasing opportunities there. We obviously also work in artificial testing systems, but we also need to make it practically relevant. We also have to move into soil environments, and this is aspects which we recently could do through innovative technology, which we lend and further develop from medical background. For example, MRI, and you can probably see on the right side the germination assays, which we can do now in high throughput in MRI, which is in soil, so we can really look how these interactions are taking place. And this gives us the opportunity to also make the interaction between the microbiome and the plants visible in a dimension, space and scale dimension, which is important for the interaction between the two domains, microbes and crops. We can do this also quantitatively. This is very important that we get really quantitative data, how the interaction actually takes place. And here you can see in that several genotypes and also some microbiome interactions between crops and during the germination with the seeds which we have in the plants. We can also do this on a bigger scale. So if we are interested in the interaction on a larger scale, we combine MRI, magnetic resonance imaging, with positron emission tomography. Which gives us, on the one hand, the opportunity to look into the structure of the root soil, the root system. On the other hand, it also gives us the immediate function, what the plant actually does using the PET, because the PET is giving us the information where carbon is short-term located and transported in the crop. So, for example, we can do this in 3 dimensions, and you can see here a corn plant. Which is growing, and you can see how the carbon is flowing in there. So the gray value is the MRI, and the color value gives us where the carbon actually flows. And we can now look into very small scale and time resolution how microbiome would interact with these activities of the crop in the same kind of spatial and time domain which is needed on the whole at that scale. We can also do this on a larger time scale of days. And on the right side, you can also see that we can quantify this data. All of these images of roots and how they are interacting with the environment and with the microbiome are in the soil. So this is real natural environment. This makes the transfer to the practical solutions much better. But obviously then in the next step, we also have to go to the field, and there are also digital technologies open a new opportunity for field phenotyping. This is including root traits. There's a lot of sensors and positioning systems. We have a lot of specific experiments. We have large-scale experiments like the one which you see on the right, where we can go into very detailed analysis of the performance of the crop above ground, but increasingly also below ground, which gives us the opportunities to get quantitative data, how the microbiome actually affects the crop performance. So in conclusion, I think there's a great opportunity here for plant microbe interaction and how they're affecting crop performance through the entire lifetime. We have methods available for seed structure and biologicals on seeds. We have methods available for the analysis and non-invasively to study non-invasively the impact of microbes on crops in a quantitative way. And we have also the ways to process high throughput analysis of microbiome impact on crops. So the interlinkage between the microbiome research and the plant research will give us much deeper understanding and hopefully also new applications, as we have heard in the past presentation from Raj. Thanks very much for your attention.
Thank you, Uli, for this very fascinating talk and insight into how we can link actually microbiomes with other disciplines to really gain more information and also make applications more feasible. I think this is very important. So thank you all speakers for these impulse talks. We will now move on with a moderated discussion. And our moderator will be Bettina Schelkle from BioEconomy. And I give the word to you, Bettina.
Thanks very much, Angela, and good morning, everyone. It's a pleasure to be here. So we've got about half an hour now for discussions. Feel free to note down any questions or any comments that you would like to contribute in the chat. Feel free to even just mention, can I have a word myself? Then I will invite you to unmute yourself and maybe switch the video on if you're comfortable doing so. And we can bring you on board into the discussion like that. I've already noted a couple of questions that have been arisen in the chat, and I'll come back to them in a moment. But what I would like to open the discussion with this morning is a question at the three speakers this morning, and what they see as the main microbiome levers within the food system, which will create the most beneficial win-win solutions to meet the Food 2030 strategy goals that the European Commission has set.
Okay.
Lene, I'm going to start with you.
With me?
Yes, please.
Thank you. I think we have a very strong focus also on health. And I think the microbiome can deliver, along with a circular approach to use of resources, that we can make prebiotic, affordable prebiotic. Some noise? Okay, I continue. Affordable prebiotic ingredients. And that would be a, no, I cannot speak across, it's too noisy.
So just check. So if anyone is currently not muted, can I please ask you to mute yourself to make sure that we don't get any background noise, please?
So I think it's the microbiome understanding both in the animal of our meat production and on the plant and in the food, in the gut of a human. It's possible to have a food where we can choose, and actually in an affordable way. It is small pieces of cell wall material, which can be such a few grams a day, can make a change in your diet. And we will broaden it to seaweeds and others, and they will have whole new types of interactions of such ingredients. So I think we have an opportunity for microbiome to be a direct stepping stone towards a broader public health opportunity.
Thanks, Lene. Brad, what do you see as the main lever within the food system that can deliver those win-win solutions we need in the next 10 years?
For me, I guess Lene has addressed the health. I'll address from the agriculture microbial tool point of view. I think from the economic point of view, the agriculture microbiome is 4.5 times bigger than human microbiome today. So human microbiome is still less than $1 billion market. You have already 4 billion agriculture microbial products in the market. Food industry, if you look at the processing, is a huge market itself in the microbiome. So if you're looking from a bioeconomy point of view, I will say the agriculture microbiome in short term will remain the main driver in this area, because it brings you the sustainability in the farming systems, environmental farming. It contributes to us mitigation of climate change by reducing the at least nitrous oxide and methane emissions from the agricultures. And if I look at that, and then you talk about the human health already, let me talk about how it's impacting the human health. For me, this is where I think the short-term gain is in agriculture microbiome. In the long term, all aspects of human society will benefit from that, as long as we have a willpower to keep going and keep resourcing the tools and the area of science.
Thanks a lot, Prat. Uli, do you have anything to add to what Leni and Prat shared?
Hard to add to all these great ideas which have been mentioned before, but I think it's very important that we will make things available to industry as well. So we have to have a sound science basis and make the science basis available to industry and to the society through products which they can actually use. That will make things happen, because the whole Green Deal is about how do we transfer our economic system. And we can only do this on a basis of, on the one hand, sound scientific approaches, and on the other hand, good products which are out there. There are already many of them, but I think it's the prospect which we have in the whole sector is much better now that we have all the opportunities from combining the different technologies, which give us the opportunity there. At the same time, obviously, there's also a risk, because as we know, and we've heard before in other talks, that technology opportunities are not always seen by society. And we have to also strongly make sure that the communication is coming up properly, and we really deliver on that scale as well.
Great, thanks a lot. Leni, you wanted to add something?
Great intervention from both of you. And I would like to add the international perspective. We are in a situation right now in Africa that there are more and more areas which are really facing agriculture, climate change challenged agriculture, lack of food, lack of water, and so on. At the same time as in Africa, just as much, they waste just as much as we do. Around 30 to 50% of all production is wasted. So we have an obligation to share this kind of microbiome and also the ingredient possibilities of improved health, for actually doing this kind of seeing it also from the ones in most need. And we can do that. And there is already in Horizon Europe an opportunity for it, but they go for classical food production in the program so far, in the call so far. I want us to try to use that as an avenue into having microbiome and more healthy food production of what is now going wasted in Africa.
Can I comment on that? Can I comment on that, Bettina?
Yeah, please go ahead.
I fully agree. And I think this is also an opportunity where we need to be developing capacities in other countries as well, because obviously, if you look about the food as well as the microbiome in, for example, Africa, and Africa is a big continent, so it's not one microbiome out there. There are many different there. So we also need to enable opportunities in Africa to do microbiome research, to do activities there, to adapt it to the conditions in other countries. So I think we should need a lot of tech opportunities there, but also adaptations to the needs in the specific environments and specific conditions where the food system is different and the environmental system is different. And we need to really make sure that this is enabled in the countries where the changes are about to happen.
Can I have a short?
Not all over, also in Asia and in other countries.
So maybe a brief comment to the group. What I would like to open the floor to is other participants within this meeting on the discussion on those win-win solutions. And Leni, I will be coming back to you in a moment again. Angela, I'm actually wondering whether you would be able to be open to comment as well, particularly because there has been a request from the bigger group.
Yeah, I see this comment. Thank you very much. I mean, I think on the one hand, we still have to be realistic also in terms of expectations. So, because we are still in an early stage. On the other hand, I think we still see, of course, the huge potential. And I think it will be most important to really address when we think about understanding, to really address natural environments, to really better understand what is going on in the field. We have to move away from what Bra called the snake oil products to really knowledge-based innovations. We have to link, of course, with other technologies, whether it's digitalization for precision agriculture, or phenotyping, as it was presented by Uli. So I think there are a lot of possibilities for completely new innovations moving in a direction of precision agriculture and precision farming, integrating microbiomes. But I think there are also great new ideas and concepts, starting from core microbiomes to syncoms and so forth, to really develop microbial solutions which are more realistic, to have really then impact, and show the proven efficacy. And I think there are a number of applications, not only for the classical crop protection or crop production problems, fertilization or crop protection, but also in regard to improving food quality. So if I think about, on the one hand, to be able to farm a broader range of cultivars, but also to improve certain metabolites, vitamins, secondary metabolites in plants, which are important for healthy food.
Thanks, Angela. And I'm actually going to follow up with a question on why do you think that a lot of these, what are the factors that hold a lot of these innovations back at the moment for them to be used on a broader scale? Is it just the issue that we don't have sufficient knowledge about it yet? Or are there other factors that come into play here as well? Angela, I was hoping to follow up with you.
Okay.
But if someone
Yeah.
someone else would like to take the floor here as well, you'd be very welcome.
I mean, of course, that's a very challenging question. I mean, I always see the issue of expectations. The expectations are extremely high, and we're, on the other hand, we have to deal here with, or we deal here with very complex systems, and so, and of course, there's always, we have to keep a balance in terms of how far do we need to move our understanding, to really be able to apply something. We don't have to understand maybe everything, or we will never be able to understand everything probably. But to a certain level, of course, we have to improve our understanding to move away from these snake oil products, yeah, and to really have more realistic and more efficient applications in place. But of course, we have already very good examples of microbial solutions which work very well. Mostly, from my understanding, I'm now focusing maybe more on the crop production side, but in terms of biocontrol, so there are some very good examples of already quite nice solutions.
Thanks, Angela. Prach, you unmuted and showed your video, so I assume you want to add.
Yeah, just few things, I guess, as you highlighted, and so does Angela, is about the technical issues that we need. As I said, that we need to both the fundamental and applied science run together. At this moment, there's a real gap both in the fundamental knowledge as well as in the applied science side of things. Expectations. I want to argue there, which industry at this moment is increasing, just tripling in 6 years? None. It's a microbiome industry and TELSA. So very few industries are growing at the rate that's the microbiome industry is growing. And the challenge for us is the expectations that everything will be fixed within 3 and 5 years. That will not happen. If you suppose that what the rate is growing at this moment, by 2030, you're talking about $50 billion industry. So I think it is already one of the fastest growing industry. And it is about what our expectation as a society is to achieve. And so it has a potential to really multiply the growth rates by several times, but it will need a time to really do long-term vision, what I meant, and to understand those sort of critical technical knowledges. And then the regulations, as Angela, some of the things has put out, so the regulations needs to move along with this as a technology coming out in the market. But I will argue that this industry is already one of the fastest industry, and we need to just make sure that it's being resourced for coming decades.
Thanks very much, Bert. Uli, did you want to add to this conversation? I saw your video popping up. Otherwise, I'm going to open to the bigger group.
Shortly add to that in a way that obviously to management of expectations is very important. And I think therefore it requires a good alignment between what industry can deliver and what science can deliver, and to have a strategy approach. And I think that's exactly the dimension which we aim at from microbiome support, to have that kind of alignment and that kind of interaction between the different sectors in order to make it, to manage expectations is also from society as well as from politicians. I think it's very important there.
Thanks a lot. Leni.
Thank you. I think, Ulrich, you are very, it's important to this to manage expectations. But on the other hand, I also have this very positive message that there are a lot of expertise growing in Africa right now. We have the platform Bioinnovate in East Africa, 6 countries organizing themselves in making better, improved use of the biological resources, working on microbes and upgrade. And we have in West Africa a fantastic culture where they have been using the fermented food, and we have microbiome studies showing that the fermented food in culture in many parts of Africa actually have a very, very beneficial effect on the both for feed and for food human health. So I think it's doable, and I think we should start developing this part as soon as possible.
Thanks, Leni. In addition to what Leni just said, for those of you who don't have a view on the chat at the moment, Fanette Fontaine has also shared the link for the African Microbiome Institute that some of you may also want to check out as another African-based microbiome research organization. From the group, would anyone be willing to or would be interested in joining the conversation, a view on what they see as a current win-win solution? Just going to give that a moment as well. I can't see anything popping up. Angela, I see that you've got your video on, and I'm wondering whether you would like to add to the conversation.
No, that was not my intention, but I can turn it off again. But maybe just a small comment on Africa or on poor soils. I mean, I think I would expect that many microbiome applications actually in regard to improving crop production will work much better in these poor soil conditions and highly stressed conditions and low input farming systems than, for instance, in let's say Europe, yeah, where we have you know, very good varieties, we have rich soils and so forth. So I think there is a lot of possibility also to you know, create small economies in regard to microbiome applications in locally to improve local crop production. Yeah? And now I will turn off my video.
Thanks, Angela. So I'm going to kind of steer the conversation into a slightly different area now, and specifically the area of AI, artificial intelligence. So one of the questions to you, Brad, earlier on was actually, how do you see the role of AI in the development of the microbial products? And maybe I'm actually going to add on to that particular question in light of what you have mentioned about the microbiome field needing major strategic alignment to ensure it delivers on the expectations that is held against it at the moment. So can you provide us your views on AI in the field, please?
So my opinion, I guess AI is already in some part, is a part of both the development, but more importantly, the performance of those inoculants in the field conditions. And I have no doubt that they will have a very important role going forward in terms of what we select as a microbial product. As I said, that we probably will be moving we are already moving from the single isolates products to the syncom, or at least in the consortium, but slowly we'll move into the syncom sort of thing. And the AI will play a huge role in going forward in the future. So my understanding is that the performance of those products is already being monitored, or at least being generated, the evidence is being generated through the AI, the performance of those sort of things. So I have no doubt in my mind the AI will play a very big, in some level, when I say the system-based approach, that's what I was also trying to say, that with the emerging tools on the robotics, you heard about the precision tools, the drone technology, the AI, all these remote sensing, all they will need to come together really for these industries to take off, particularly in agriculture, where we are talking about a vast area. Human monitoring the humans is a little bit more constrained because doctor and GP can monitor, but agriculture, you're talking about vast areas across the globe. So all these tools needs to come together really for these tools and technologies to take off. So I have no doubt that the AI will be one of the critical components moving forward.
Thanks, Pra. And then to my final question to the group, before I will be asking our rapporteur to close up the discussion, seeing that we are starting to come towards the end, is based on one of the questions that we've received through the chat as well, and that's specifically what is the role of microbiomes in biomass production for material use, so in non-food. I would quite like to make the link back into the food system, seeing that this is one of our main topic areas within this group, by asking you to also consider, for instance, how waste streams from the food system can be used and how that might link to microbiome and products that then can be used in non-food areas. Anyone happy to take that particular question and share their opinion and thoughts on it? Brad.
Okay, so there is already a lot of things that is at this stage. I will say that research development stage. So the people have used a lot of things in nursery for the plant biomass productions, taking into the field and looking at how the field things is. I'm a part of a recently funded projects in the New Zealand that looking at the pine plantations. I know there is quite a significant research going in the USA in the bioenergy crops. And biomass productions in this. Those are the developmental stages, not just the research in the lab, but in the field conditions. I'll give you an example that you talk about using the waste. There is a company in Australia called Nutrog, and it's a good sized company. It's have annual sales around $15 to $20 million per annum, and they use the chicken waste and convert them to pellets. And they can't, at this moment, not able to supply enough to the farming communities in the use of agriculture. So there are opportunities and there are people doing those sort of things. That is it. Where I see and some of the things people highlighted in the charts, so like Anne has highlighted the policymakers, somebody highlighted about the regulations. What we need to is to bring together those stakeholders in one room so that we can promote, support the translational research as well as the commercializations. And as I said, that there's a lot of multiple areas like manufacturing, you take an example, the regional economy, environmental outcomes, and environmental industry, in fact, that will benefit from this sort of tools and technologies coming in the market. We just need to bring these peoples together, inform the policymakers, get the regulators on the boards, and move these things faster a bit more.
Thanks very much, Praj. I think Ila's comment in the chat is also very valuable here, that some of the products that are coming to market take more than 6 years because of the registration pathway that is involved there. And certainly if that sped up, a lot of the application of the microbiome products would come forward as well. Yeah. Lene.
This is a very big field, but I think the most important is that we start finding out what is the kind of waste which can be upgraded to food and feed. Because now waste is most of the processing, but much of it from non-food can also be food quality because it was never dirty in any sense. So that's the first part. The other part, there is, for instance, a fantastic project where I've been involved as well, is where we take the entire microbiome on one of the most fly types of fly larvae, which are the best of all to eat different types of organic municipality waste, and to use the whole microbiome of that fly, and make a fermentation and make that more and more to a more kind of reduced complexity microbiome, but still very efficient. So there are so much to be found there, and I think, but first of all, let us distinguish between what we can upgrade and what we should downgrade to make to interesting soil improvement and other types of factors.
Thanks, Lene. I just see in the chat as well that biogas has been mentioned as well as one of the potential non-food products that come out from microbiome applications.
But Bettina, this is the important part, because biogas is the only one which is subsidized heavily in Europe, which means that it attracts a lot of biomass which has potential for upgrade. But what we can do is to make biogas upgraded to other types of product than just biogas. And the anaerobic digestion microbiome can actually be used also for a phosphor up concentration for a new type of fertilizer. Waste is gold.
Thanks, Lene. We need a lot of gold for all of the alignment that's required in the microbiome field. Uli, we've got about 6 minutes left, so I'm going to ask you to keep it short before we pass it on to our rapporteur.
I think it's what we observe there is something which we see in the general bioeconomy also as a aspect there. Because we are forming new networks of interactions between different sectors, food, waste, non-food materials, we are touching very different parts of law and registration, which have been developed from very different backgrounds, and now through the bioeconomy we interlink them in a way which is not foreseen by these law systems, and that's the tensions which are developing there. At the same time, this also generates opportunities. So if we move from an energy field to a chemistry field or food dimension, this is opportunities where, for example, the energy sector, just like Lene just said, could benefit from getting much higher value products than just burning gas in a pipeline, but really generating food, for example. And that's an opportunity. At the same time, it's a challenge because we are moving from one law system into another law system, which are not compatible.
Thanks. I think that's an important consideration here as well. I would also just want to acknowledge a couple of points that I've seen popping up in the chat, and that's the fact that to manage expectations, we also need to educate, and that requires microbiome literacy through all of the ages. And we've got one other non-food application, which is basically alternative proteins from mealworms from vegetable waste. This is leaving me to hand over to Tanja Kostic now from the AIT Austrian Institute of Technology, who will basically capture the last half an hour in hopefully just a few sentences before Angela is wrapping up. Thanks, Tanja.
Thank you, Bettina, and also from my side, thanks to all speakers and all participants of discussion, both online and in the chat. I will try to do my best, but I think I have identified few points. The first really interesting point for me was that there was sort of a general agreement that we need more this holistic and systemic approach in different, starting from microbiome is more than bacteria, going over that we need to consider plants and animals and also humans in this holobiome principle, that we need to consider the whole food system and how different parts of it influence each other, also linking them ultimately to One Health. But I also found interesting this link that we need to start to think more globally and to start to enable people in different regions to also be able to learn from what they have from their natural resources, be it now environmental or food. I also saw there was a lot about the interlinking of new and different technologies. So we were mentioning AI, we are mentioning robotics, we are mentioning new sensoric technologies and how these could be also brought or pulled together to push microbiome R&D forward. I also found really interesting and important discussion about managing expectations and about importance of linking different stakeholders from science and providing this sound science base to the industry to enable them to provide these applications, but also importance of involving policy and putting up the framework. And I also noted what you now said at the end, this education is a really, really important part of it. What I would like to say as a wrap up of my sort of my summary, I would just like to quote Bry in what he said, long-term vision rather than a short-term outcomes. I think this is really the solution for what we are trying to do here. And I also think this is where I'm really happy to be a part of microbiome support. And I think we started on the right track to it. So thank you all. And if I missed something, please feel free to push it to me in the chat.
Yeah, thank you, Tanja, for this very important final statement and for the summary. Thank you, Bettina, for the excellent moderation, and thank you all for your contributions. I think they're all very valuable. It's very important to have these kind of discussions in order to really take the right way into the future. Our workshop is not finished. We will have the second half of the workshop will be in the afternoon, yes, it will be more on discussing the gaps and needs related to microbiome-based research and innovation, more on the policy regulation side. But I think we are open what we will discuss. We will continue our discussions. We will also have again 2 talks in the afternoon. I hope that the link will be the same. In case you cannot access, please send me an email and I will try to send you the right link in case I receive another link, or I don't know, we will see. So I hope we can see you all again in the afternoon, and wishing you some nice break until 4 o'clock in the afternoon. Thank you very much. Bye.
Thank you. Bye-bye.
Bye-bye.
Bye. Thank you.
Bye, thanks.
Thank you, Angela. Bye.
Thank you
Bye,
very
Brad.
much.
Ciao.